Good morning, students. I am Professor Nitesh Sharma. Today, I am teaching you genetic engineering. This topic, genetic engineering, is included in the paper second of second year genetics and biotechnology. Students, as you all know, that the word genetic engineering is related to the engineering with the genes. So, genetic engineering, also called genetic modification or genetic manipulation, is the direct manipulation of an organism's genes using biotechnology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. New DNA is obtained by either isolating and copying the genetic material of interest using recombinant DNA methods or by artificially synthesizing the DNA. A construct is usually created and used to insert the DNA into the host organism. The first recombinant DNA molecule was made by Paul Berg in 1972 by combining DNA from the monkey virus SV40 with the lambda virus. As well as inserting genes, the process can be used to remove or knock out genes. The new DNA can be inserted randomly or targeted to a specific part of the genome. So, students, now you are able to understand what is genetic engineering. An organism that is generated through genetic engineering is considered to be genetically modified, and the resulting entity is a genetically modified organism. The first GMO was a bacterium generated by Herbert Boyer and Stanley Cohen in 1973. Rudolph created the first GM animal when he inserted foreign DNA into a mouse in 1974. The first company to focus on genetic engineering, Gentech, was founded in 1976 and started the production of human proteins. Genetically engineered human insulin was produced in 1978 and insulin producing bacteria were commercialized in 1982. Genetically modified food has been sold since 1994 with the release of the Flavor Sav tomato. The Flavor Sav was engineered to have a longer shelf life, but most current GM crops are modified to increase resistance to insects and herbicides. Glowfish, the first GMO designed as a pet, was sold in the United States in December 2003. In 2016, salmon modified with a growth hormone were sold. So, students, genetic engineering is the very important branch of life science. Genetic engineering has been applied in numerous fields, including research, medicine, industrial biotechnology, and agriculture. In research, GMOs are used to study gene function and expression through loss of function, gain of function, tracking, and expression experiments. By knocking out genes responsible for certain conditions, it is possible to create animal model organisms of human diseases as well as producing hormones, vaccines, and other drugs. Genetic engineering has the potential to cure genetic diseases through gene therapy. The same techniques that are used to produce drugs can also have industrial applications, such as producing enzymes for laundry detergent, cheeses, and other products. So, students, genetic engineering is the indispensable part of our routine life. The rise of commercialized genetically modified crops has provided economic benefit to farmers in many different countries, but has also been the source of most of the controversy surrounding the technology. This has been present since its early use. The first field trials were destroyed by anti GM activists, although there is a scientific consensus that currently available food derived from GM crops possesses no greater risk to human health than conventional food. GM food safety is a leading concern with critics. Gene flow, impact on non target organisms, control of the food supply, and intellectually property rights have also been raised as potential issues. These concerns have led to the development of a regulatory framework which started in 1975. It has led to an international treaty, the Cartagena Protocol on Biosafety, that was adopted in 2000. Individual countries have developed their own regulatory systems regarding GMOs, with the most marked differences occurring between the US and Europe. So, students, now you are able to understand the concept of genetic engineering. This is very important and an indispensable branch of life science. Thank you very much.